Alright, so let's take a look at chapter number 27, which is all about prokaryotes. And remember that in the last chapter we said that the prokaryotes used to be the kingdom Monera, but were divided into the domains bacteria and archaea. So one of the things that you're going to see in this PowerPoint is the differences between the two groups of organisms. So, general motto for prokaryotes, we've seen this before in cells, what makes up a prokaryote. Um, Obviously, they have a plasma membrane. All cells have plasma membranes. All cells have DNA, but one of the differences between prokaryotes and eukaryotes is that their DNA is not in a nucleus. Bacteria also have a cell wall, but unlike with plants, their cell walls are made out of proteins, specifically something called peptidoglycan. Remember that plant cell walls were made out of cellulose. On the outer extremity of some bacteria, they have capsules, which are basically an extra layer of protection, and it's usually useful, especially in extreme environments of, let's say, extreme heat or salinity. And then you also have prokaryotes with pili on the outside. That's assuming that they have an F plasmid. Remember, that's the fertility plasmid, which allows them to undergo conjugation, which is the transfer of one plasmid from one bacteria to another bacteria. So pili are very important in, remember, the horizontal gene transfer that we talked about. That's one of the ways, remember, that bacteria, because they're asexually reproducing organisms, that's one way that genes can spread throughout the population. And it's also one of the reasons why we have such problems with antibiotic resistant bacteria, which in the lab that you guys are looking at right now, you can see that there's bacteria still growing on erythromycin plates, for example. Alright, so they also have flagella. Remember though that their flagella are different than eukaryotic flagella in structure, although their function is the same. They help propel bacteria and help them get around. Alright, so we've already talked about how the kingdom Monera no longer exists, that we now have the domains bacteria and archaea, and that in each of those there are multiple kingdoms, but there's a lot of debate right now about what should be the delineations of those kingdoms, largely based on genetic evidence. We do find that bacteria tend to have, I should stop saying bacteria, prokaryotes have three shapes. Cauci, which are spheres, and you can see these down here in this electron microscope image. You've also got the bacilli, those are a rod-shaped form, and then you've got the helical bacteria, which are in the shape of a spiral. You can see right here an example of an organism that is a helical shape would be the bacteria that causes syphilis. Alright, so there's structural characteristics. What makes up a bacteria? We've already seen the general idea, but remember that one of the things that separates them is the cell wall. And the bacterial cell wall, as we said, is made up of proteins called peptidoglycan, and that's what separates their cell wall from those of eukaryotes. There are two different types of bacterial cell walls. There's one type that you can see down here in the bottom left hand corner, and this is showing you a plasma membrane, and then on top of the plasma membrane there's a peptidoglycan cell wall, that big brown chunk there. These type of bacteria are called gram-positive bacteria, and the reason why they're called gram-positive bacteria is that when we put them into a stain, or into a stain called Gram stain, obviously made by a scientist named Gram, their color becomes this dark purple, and that's because the stain is able to attach and fix to the peptidoglycan. And so when the stain comes into contact with the peptidoglycan, it forms a purple color, and so we call them gram-positive bacteria. These are the type of bacteria that are susceptible to a greater spectrum of antibiotics. Having only one cell membrane makes it more likely that the antibiotic will be able to cause its effect. There are another group of bacteria that are called gram-negative bacteria, and these bacteria have basically two cell membranes. So you've got an inner membrane down here below, and you've got an upper membrane, and these two membranes sort of are what do you call it, interspersed by this peptidoglycan layer. So there's a thin layer of peptidoglycan, but it's like sort of like a piece of ham in a sandwich, with the bread on top, and on the bottom sort of covering it. Now, these bacteria are called gram-negative because this outer membrane protects the peptidoglycan from the gram stain. So the gram stain isn't able to bind with the peptidoglycan, and so they show up as a light pink color here on our microscope image. The thing about gram-negative bacteria is that there's fewer antibiotics that will work against them because this outer membrane can act as a selectively permeable barrier, which means that not all antibiotics are able to make it through the membrane and cause their desired effect within the bacteria. So they are a little bit more difficult for us to get rid of. But there are different antibiotics that can be used against gram-positive and gram-negative bacteria. Just this outer membrane is something that makes it more difficult to have its effect. We've also saw in that first slide that there was often a capsule around a bacteria, and what those capsules can do is basically help them attach to other bacteria, form colonies, and it also serves as an extra layer of protection against high temperatures or high salt concentrations. On the outside of bacteria, we can see here there's also these pili, these extensions that allow for conjugation between two different bacteria, so they can transfer their plasmids from one to another. Alright, so how can bacteria get around? One way that they can get around is by using their flagella. It spins and swirls just like a eukaryotic flagella would, but the structure is different, and they also have a helical shape, some bacteria. We saw in the first slide are spiral-looking bacteria, and I said as an example, there's syphilis, and the way that syphilis can move around is, I don't know, you kind of combine it like a slinky-type snake, that by the movements that it makes in a spiral, the contractions and relaxations, it's able to get its way around. So that's another way a bacteria can move if it has that shape. Remember that we call the helical bacteria the spirochetes. And then you've got also slime. So some bacteria can release different types of liquids that basically act as, what do you call it, like a lubricant that would help them get around. Remember that two dry things rubbing against each other, there's going to be friction, it's going to be difficult for them to move around, but with the secretion of that slime, they're more able to do it. And you'll remember from our talk on ecology at the very beginning of the year that taxis is a movement away or towards a stimulus, and bacteria exhibit many different types of taxis, for example, towards light. If we're talking about photosynthetic bacteria, how does a small tiny organism find food sources? Well, it's able to move its way towards, increasing concentrations of, let's say, something like glucose so that it's able to find its food source and survive. So bacteria have a bunch of ways of getting around by flagella, using their helical shape, using a slime, and often these bacteria aren't just aimlessly moving, they're often moving in the direction of something.